Okay, so you now know what the neuron looks like, and we've talked a little bit about how it might function in terms of it actually is responsible for sending on electrical signals. But the question is, how does it actually perform this task? How does it actually send electrical signals in the first place? In order to understand how it does this, we need to understand something called resting membrane potential. And resting membrane potential is not unique to neurons. It's actually there is a resting membrane potential in all cells, but neurons are able to fluctuate this resting membrane potential to send their messages. Let's have a look at how they do this. Well, what a resting membrane potential is basically a difference between the charge of the inside of the cell compared to the charge on the outside of the cell. And you can see here that we've got two electrodes, one inserted into the cytoplasm and one inserted into a region just outside of the neuron, hooked up to a machine that's measuring the charge or the voltage. And you can see that when you measure the inside to the outside of the cell, it comes up with a value that is minus 70 millivolts. And this is what's said to be the average resting membrane potential of a neuron. Minus 70 millivolts, that basically means that the inside of the cell is negative compared to the outside of the cell. So at rest, the inside of the neuron has a negative electrical charge relative to the outside, and this is because there are more negatively charged particles, or ions. Remember that back from the first couple of weeks of this unit. There are more negatively charged ions on the inside. I'm going to talk about in the next slide how this actually occurs. We've said that the difference between the inside and the outside and the charge is called the resting membrane potential, but because it does have a charge difference, we call the cell polarized. So the cell is said to be polarized simply because the inside and the outside have a difference in charge. So how do we actually set up this resting membrane potential? Well, there's a couple of ways that this occurs. So what we've got here is a picture of the inside of the cell compared to the outside of the cell. And then obviously here we've got our cell membrane showing our phospholipid bilayer here and here. And then we've got a number of channels. We've got potassium channels shown in green, and we've got our sodium channel in blue, and the sodium-potassium ATPase pump. So you'll remember all of these things from what we've discussed early on in the semester. Okay, so how is it that we come across a resting membrane potential that's negative? Well, the first reason this occurs is because inside the cell, in the intracellular compartment, we have a lot of negative proteins. So I'm going to draw these in yellow. Negative proteins. Okay, so negative proteins on the inside of the cell. And that's the first reason why the inside's more negative, because we do have negative proteins and also some DNA. They both have negative charge associated with them. But in addition to this, we also have lots of potassium on the inside of the cell. So you can see here, lots of potassium sits on the inside of the cell. So we'll put lots of K+, K+ being the chemical way to write potassium. Lots of potassium on the inside of the cell, which doesn't mean there's no potassium on the outside. There certainly is, but there's less potassium on the outside than what there is on the inside. Now let's compare that to what goes on the outside of the cell. You can see here that it says there's lots of sodium. On the outside, or the extracellular compartment, there's lots of sodium ions, and far more sodium ions on the outside than what there are on the inside. In addition to that, on the outside, we also have a number of chloride ions, trying to balance up these positive sodium ions. So you can see there's a relative difference in the types of ions on the inside and the outside of the cell, and this is the second reason that we have a resting membrane potential. So we have a difference in the ions in the extracellular and intracellular and intracellular compartments. Okay, so you might be thinking, well, how does this all actually mean that we're negative on the inside? There might be a difference in the concentrations of these ions, but they all seem to have positives. Well, there's a third reason why we have a negative charge on the inside of the cell, and that's simply because of the availability of ion channels to actually pass these sodium or potassium ions from the inside to the outside. let's have a look at this. You can see we've got, as I said, the potassium channels in green. And these are leakage channels, which means that they can pass potassium from the inside to the outside of the cell down their concentration gradient. So you can see that there's lots of opportunity for potassium to leave the cell. On the other hand, we have a sodium ion channel here. And this sodium ion channel again is a leakage channel, meaning we'll have a concentration, sodium ions going down their concentration gradient, this time from the outside to the inside. But you can see there's fewer sodium channels than what there are potassium channels, which means there's less sodium entering the cell than what there is potassium leaving the cell. So we've got lots of potassium leaving the cell, so lots of positive charge leaving the cell, but less positive charge entering the cell, kind of contributing to our negative charge. To make matters even worse, we've got this sodium-potassium ATPase pump. And because it's a pump, we know that it's going to pump our ions against their concentration gradients, and it's going to do so with the help of ATP. So in this situation, the few sodium ions that did come into the cell, well, they're going to get pumped back out. We've got a sodium here, here, and here. So 3 sodiums are getting pumped back out of the cell against their concentration gradient, but you can see there's only place for 2 potassium ions. So 2 potassium ions are coming in, whereas 3 sodium ions are being pumped out, therefore meaning that the inside is even more negative to the outside because there's fewer positive charges there. So our third reason that our inside is more negative is because of the transport of ions, or positive ions in particular, positive ions across the membrane. Remembering that that's because of the additional availability of potassium channels compared to sodium channels, as well as the activities of the sodium-potassium ATPase pump. So if all that sounds a bit much, the biggest thing to remember is that the outside is relatively positive compared to the inside, which is relatively negative, and the difference is minus 70 millivolts. And you can see that here. So we've got our inside cytoplasmic region being more negative than our extracellular compartment. And that's why when you put an electrode inside the cell and compare it to the reading of an electrode outside of the cell, you get a reading that's minus 70 millivolts. And that's what we call resting membrane potential. And when we alter that by either increasing or decreasing it, we can actually start to communicate.